appreciate it. I got to do a little bit to get everybody on board. All right. We're going to get started a mere 3 minutes late. This is a record for me. Thank you for coming to the last scheduled event for the 2016 Maine Science Festival. This is a showcase event for Sunday. Showcase for us means that we do it every year. So for us, every year, that's 2. Pretty good. We've had this twice now. There's a couple of things about this session that you should know. Number one, all the panelists here are, and I'm going to say this, they don't like it, they are rock star scientists in what they do. You know, they are remarkable in their work, really innovative stuff. Some are on the lower end of the, earlier end of the career path. Some are a little bit higher up. All do really great stuff. The secret, I will admit here today, this is my favorite session of the Science Festival. This event, if we only had this for the Science Festival, I would be happy. So you're in for a real treat today. So this is how this works. They will have 5 minutes to explain their work. You know, the stuff they've been working on for 10, 15, 20, 30 years. 5 minutes, summed up. We will cut them off with a cowbell. They know about this, so. Then it's 5 minutes of questions. So I'm going to try to moderate the questions for you, so just raise your hand if you have a question. Since we only have 5 minutes of questions, it would be really helpful if you got right to the question. That would be really great. So, once we get through with everyone, we have a reception afterwards. Please join us. That's your chance to actually have a little bit more in-depth conversation with these phenomenal researchers and scientists. So, without any further ado, I'm thrilled to introduce Vivek Kumar, who's an assistant professor at the Jackson Laboratory. Vivek, okay, so here's the other thing that you get that others don't. I'm going to totally explain with a very long bio why these guys are here for 5-Minute Genius. So bear with me. It's totally worth it. Vivek received an AD in biology from the University of Chicago and earned his PhD at the University of California, San Diego, and joined the Jackson Laboratory in 2014. Vivek's lab studies neural circuits in the brain whose misregulation leads to behavioral abnormalities, including addiction, attention deficit and hyperactivity disorder, and depression. Using mouse molecular genetics as a foundation and a combination of biochemistry, physiology, and imaging techniques, Vivek's lab dissects these complex behaviors in mammals. Using 2 functional genomics approaches in mice to identify genes and pathways that regulate these behaviors, this is a powerful and unbiased forward genetic approach that makes no a priori, did I say that right? Yep. Assumptions, and only require a clear, well-defined assay for gene discovery. This is all going to make so much more sense once he talks. Vivek's lab uses a high-throughput screening pipeline to discover mutants for cocaine response and open field behavior. Using physical mapping followed by next-generation sequencing, they have identified novel genes and alleles? Sorry. Thank you. This is getting better and better. That regulate cocaine response and anxiety-related behaviors. Please join me in welcoming Vivek.
Thanks, everyone. So I'm going to go fast because I just had a bottle of water. So I'm going to tell you about modeling addictive behaviors in mice, or how we're using mice to study this complex disease. So this is a quote, and I'll buy someone a bottle of water if they can tell me who said this. With cocaine, one snort, and it owned me, body and soul. Click. Cocaine was my on switch. Anyone? Except my daughter. Stephen King, a famous Mainer. This is Paul Erdos, another bottle of water, if you know who this person is. If you're a mathematician, you'll know who he is. He's like the Kevin Bacon of mathematics. He used to be a hermit. They have these degrees of connectivity to Erdos. He was a hermit mathematician who for years traveled, and he would go to different spots and collaborate, publish with them. He, for 30 years, was using methamphetamine. And his methamphetamine use concerned his friends. So one of them actually bet him, said, if you can quit, if you can quit methamphetamine for 1 month, I'll give you some money. So he did. He quit for a month. He didn't use any drugs. And then at the end of that month, he said this, which is, you showed me I'm not an addict, but I didn't get any work done. You set mathematics back 1 month. mind telling you this. So this is a fundamental problem we're trying to study, which is most of us are going to use a drug with some abuse liability, addictive substance, some point in our lives. But a few of us are going to escalate and go use it uncontrollably and are going to become addicts. What controls this transition? Why are some people casual users and other people become addicts? And genes, we know, play an important role, as does the environment. And this is a chronic disease known as addiction. So we use mouse model systems to model this, model systems to study this. And mice turn out to be actually good models for cocaine use. So this on the left is a mouse that does not have cocaine. On the right, you have a mouse that's been given cocaine. We just inject it with a dose of cocaine. And you can see, just like in people, it induces hyperactivity. And then mice will actually work and they will self-administer the drug. They will do some work to get the reward. So I'm going to tell you a story. 1951, Harry Truman was the president. Yankees beat the Giants. And Gene Kelly and this movie, An American in Paris, won 6 Academy Awards. But for us mousers, something really important happened. We had a colony of mice that were shipped from Bar Harbor, Maine, from the Jackson Laboratory to the National Institute of Health in Maryland. Now, these mice that were shipped were exactly identical. They were clones of one another, no genetic difference at all. We took half of them and kept them here. The other half went to the National Institute of Health. I'm going to call these guys the Maryland mice. And these Maryland mice in 1951, so they're exactly identical. But it turns out in 2014, when you look at cocaine response, this is measuring cocaine response in Maryland mice and in Maine mice, the ones at the Jackson Laboratory. The Maryland mice now respond less to cocaine at multiple dosages. So genetically they were identical, but now there's a difference in their behavior. So I'm going to go fast, but as a geneticist, we want to study these differences, and these strains were identical. So what's the difference that causes this behavior difference? So we use genetic mapping and sequencing and all the tricks of modern day molecular biology, molecular genetics, and we go from 2.8 billion nucleotides to one nucleotide, and we found a mutation in a gene called cyclic 2. And this mutation regulates the difference between the two in terms of the phenotype. So this is like taking two populations of India, which is roughly around 2.5 billion if you 2x, and going to find a single person in that. So this is a massive task. It took about 3 years to do this. So what does this gene do? And here we get into circuitry. So this gene, this is a mouse brain, not a human brain. This gene controls circuitry in the brain. And we have these neurons in the brain called medium spiny neurons. And I'm going to show you a picture of one end of these neurons. And this is that picture. And this gene is controlling the shape and the structure of neurons. And what I've shown you here is just a microscope slide. This is the data that I collected here. And it's actually controlling these little protrusions where chemical signaling takes place in the brain. So, question, what controls this transition? Here's the observation.
No, we're cutting you off. No. Which is great because it's question time. Thank you. No, keep it up. You can put that one up. Back up the one slide. We'll keep it there. Do we have any questions for Vivek?
Could you finish
Yes.
the end?
Go ahead, real quick.
So we start with this question, which is we want to find genetic differences that kind of make one person vulnerable. We are using these mice. And here I showed you a single nucleotide change out of 2.5 billion nucleotides that makes the Maryland mice less susceptible to addiction than the Maine mice. And so mutation in Maryland mice occurred. We can tell when it occurred. And then we use neuroscience techniques to really dig into mechanism afterwards.
The Washington effect?
The Washington effect.
Great
They'd be using more drugs.
Yes, sir.
Were these mice in Maryland being bred with non-cloned mice over those?
No. So the colonies were kept what we call, they're essentially mated with each other. So these differences have arisen through genetic drift because replication of DNA is not 100% accurate. There's always some variation that gets picked up in every generation.
Yes, sir.
Were there different environmental factors with this population?
Sorry, ask that again.
Different environmental, different
Yeah,
temperature,
so this is one of the advantages of using mice. We can control their environment really well, which is something we can't do in humans. So in this case, with the data I showed were mice that were raised in identical environments. We basically got them from place A to place B, raised them in our colony, so we control the environment very carefully. But that's a great question. Yes?
Do you ever check on what the impact of the drug would be on the in vitro and a baby mouse by looking at introducing the drug later?
Yeah, like in utero.
Yeah.
In this case, we have not, but that's an interesting approach. Like, how would this gene affect development of formation of the circuit during development? Yeah. Sorry.
No, you go ahead.
Yeah.
You're total. This is great. Yep.
Do you have any problem from inbreeding with these? You start with 8 or so
Mhm.
that are identical, and then they inbreed generation after generation.
Right.
No problem.
So these mice are actually inbred from the very beginning.
Yeah.
So that's one of the reasons. So when C.C. Little started the Jackson Laboratory, the big contribution that these mousers made was creating inbred strains of mice, because that allowed us to get rid of kind of the genetic noise. So you start with mice that are completely clones of one another. So it wasn't that they were inbred after they were shipped. They were actually inbred before they were really shipped.
So does this research also generalize to other drugs which work as depressants, like cocaine?
Yeah, so this gene, but specifically Cyphic2, since we made the discovery now, people are looking into whether this, actually here at the University of Maine, we're collaborating to look at alcohol, to see if this mutation in this gene affects alcohol. We're collaborating with more researchers looking at nicotine. So we're so people are now starting, once you give them a gene, they can kind of go further and dig more into the mechanism of their favorite drug.
Do you think there's an alcoholic gene?
An alcoholic gene, no. And we can talk about that over.
And they will be there.
Yes.
I just have a question. If they're all genetically identical, why is there just between Maine and Maryland if the environment's exactly the same?
So it has nothing to do with the environment. It has to do with the genetics. They were identical, but over the course of, in 1951 they were identical, but between 1961 and 1974, the Maryland mice picked up a mutation, and that mutation is in this gene, and that mutation leads to this lower response.
So the Maine mice also had genetic drift, but just
Yes, but not this one mutation. Yeah, absolutely.
Right in back.
How do you know if you're using, if you're doing other experiments, how do you know genetic drift is affecting your outcomes if you think your star quality?
Yeah, so that's something as a geneticist we always have to worry about. Most of our experiments are done in 1, 2, maybe 3 generations. But if I take a mouse colony and I keep it and I breed it for 10 generations or more, then I actually have to designate it as a mouse colony in my lab. And then there's a whole nomenclature that goes behind it. So we have methods of keeping track of that. And we also have methods of actually preventing genetic drift. And I can explain that more to you also.
You've already done. Anybody else? He's my son. I can do that to him. Go ahead. Go ahead.
Have you seen any virus in cause mutation in animals?
Viruses.
Viruses.
Yes, so there are not just viruses, but also retroviral endogenous viruses that are known as retro elements in the genome that can lead to mutations. So certainly when we start with this phenotypic difference, we don't know what can cause it. It could be some other kind of structural variant that's caused by the principles of house. Or I don't think it could be viruses, but in this case, it's not.
All right. That was excellent.